Okay, so the first disease we don't have in Utah that I want to mention is bacterial scorch. And for those of you that do tree work in Utah, I'm sure you're familiar with the water-induced scorch that we see every summer on a lot of different tree species starting late July or so. And that's a result of that imbalance of water from the root system to the leaves. The leaves are transpiring, the weather is hot, it's dry, but there's not enough water coming up from the root system to support the lost water from the foliage. And so you get this browning of the foliar tissue. So this type of scorch is very common in Utah. We see it all the time. But bacterial scorch is different. It's a disease caused by a bacteria called Xylella. It says here, not yet identified in Utah, and actually this should be not yet identified in Utah on oak trees. We actually have seen it in southern Utah on the tree called catalpa, but in northern Utah, we've not seen it on oak trees. So that's what should be written there. And it's spread by insects that feed on phloem, like spittlebugs, leaf hoppers, tree hoppers, but the exact species is not known. And this particular pathogen has a wide host range. So I mentioned the catalpa in southern Utah, oaks, grapevines, alfalfa, so very wide, diverse host range. So initial symptoms will appear as a chlorotic area, and could be in the middle of a leaf or on the edges of the leaves. And then that chlorotic area will die, turn brown, and these types of symptoms will spread in the tree. So here you see the symptoms on elm at the top, and maple, box elder, sweet gum at the bottom. So what I'm really looking for is this disease on oak in Utah. I know there's not a ton of oaks, but it's still something to look for. And if you look at the pattern or the distribution of the brown or necrotic tissue on the oak foliage here, even on the one branch shown, not every leaf is affected the exact same way. This leaf is fully green, and then there's some leaves that are half dead, some that are completely dead. And then elsewhere in the tree, it's not, you don't see any symptoms. So this is what you might see with bacterial scorch, where maybe just one branch is affected, or just a few leaves on one branch, or just one side of the tree. But with the water-induced scorch that we see all the time, it's pretty evenly dispersed in the tree, and the symptoms on the foliage is almost, if you folded a leaf in half, it's almost a mirror image on one side of a leaf and the other. So this is just a stack of oak leaves that have this water-induced scorch symptoms, and same with on the right. So back to bacterial scorch. This picture on the right, you can see it's just affecting one side of the tree, but leaves will drop to the ground, the crown will thin out, and eventually they'll be dieback and decline. And the symptoms reappear year after year. So it's almost like a systemic infection. And so areas where this does occur, which is all of eastern US, and again, I'm talking about on oak trees, and some parts of the West, what they're doing to manage it is, there's not a lot that can be done, but they're pruning out the symptomatic branches, anything dead. And they're working on using some injections of an antibiotic called oxytetracycline. It's not a cure, but it's shown to delay those symptoms, and it has to be reapplied. And also they're considering, well, let's just replace these trees because the symptoms do come back year after year. And so this list here are trees that are resistant to bacterial scorch. Okay, so the next disease we don't have in Utah, but to keep an eye out for is pine wilt. And this is caused by a nematode. And just like the bacterial scorch, it needs to be introduced into the tree by a vector. and the vector is pine sawyer beetles. All right, so this webinar series is invasive pests, but this pine wilt nematode is actually native to the US. But it's an emerging pest because we plant a lot of non-native hosts as ornamental trees, which is fine, but the pinewood nematode has co-evolved with our native hosts. And so moving to the non-native host, then it's causing this more severe disease. So trees that are affected, they wilt, they turn brown, and they die in as little as 3 weeks. So I don't think I have an image of the United States in this presentation, but the closest that the disease has been diagnosed in the US is in parts of Colorado. And I believe it's been found in Montana as well. But our ornamentals that we plant often that are highly susceptible are Scotch pine, Austrian pine, and Mugle pine. Our native white pines can get it, but typically don't see the symptom of expression of the disease as these non-native trees do. And I'm sorry, I forgot to mention what I'm going to do with questions. So you guys feel free to type your questions in chat, and I'm going to address them at the end, unless it's what I'm talking about right at the moment. So feel free to ask your questions and we'll get to them at the end. All right, so the life cycle of the pine wilt nematode. The vector, as I mentioned, is a pine sawyer beetle. And we have this white spotted pine sawyer in Utah. So it does occur here. It looks a lot like the Asian longhorn beetle. So we get a lot of look-alike submissions of this beetle. So in the spring, when the beetles emerge from their pupation, they do what's called maturation feeding, where they're feeding on the foliage of succulent shoots for nourishment. But they may be carrying the nematode in their body. And as they feed on the succulent healthy tissue, they're introducing the nematode into the tree. And so as a result, the nematode then spreads throughout the resin canals, feeds on those, and eventually plugs them up. There's no flow of water up and down the tree in the vascular system, and the tree dies pretty quickly. As the tree dies, other insects come in and they introduce different fungi. And one of them is called blue stain fungi. So a lot of our conifers that are dying will have this fungi inside them, and the nematodes also feed on that blue stain fungi. So they are reproducing like crazy in these dead trees. Now, back to the beetle. So the beetle, to lay their eggs, they prefer dead and dying trees. And so they're going after these trees that may already have the nematode circulating through them. They lay their eggs, the larvae feed in the tree, then the adults emerge carrying the nematodes, and the cycle repeats. So this is just another couple of images of what to look for with that wilting, foliage, fading, and then, like I said, a pretty quick death. The size of the pine sawyer beetle is pretty big. So just from head to abdomen is about 1.5 inches or 1.25 inches. And then of course, with the long antennae, almost the same size of the body, then you got about 2 to 3 inches. So a decent size. All right, so where this occurs, and as I said, it's in Colorado. So we have not detected it in Utah, but we're keeping an eye out for it. It's managed by removing the trees quickly, because the dead and dying trees have the nematodes inside them. And I wanted to point out that the nematodes actually can be seen in a microscope by taking chips of a wood from a suspected infected plant, letting it soak in water for about 24 hours, and then looking at that, taking a sample of that water and looking at it under a microscope. And that can be a way to identify if the nematodes are present. So infected trees, removal, firewood. Of course, the sawyer beetle may lay eggs or be emerging from the firewood from infected trees. So those should not be kept around. If they're chipped or burnt or chipped, that's okay, or burning. And then some have experimented with injections of the insecticide abamectin, and that's for the pine sawyer beetle. But there's really no indication that it's a recommended cure-all for or prevention for this particular disease. Okay, so now I'm going to switch to some diseases that are already present in Utah, but in some cases we are starting to see more of them. So one group are the diseases called Phytophthora, and there's a pronunciation there for you, Phytophthora. So these are somewhat a mix of native and introduced species that occur in our soils in Utah and will only cause disease under the right conditions with the host and with the soil conditions. So it's a mix of different Phytophthora species and a very wide host range in terms of our ornamental trees and shrubs. There's lots of fruit, berries that are susceptible, vegetables that are susceptible, but here's some that I have seen in Utah. So for the life cycle, it overwinters or survives for many years in the soil as a resting structure that may be living in the soil, or it may be living in plant debris from a prior infection. So that the pathogen that's in the soil forms a special structure called a sporangium, and inside the sporangium forms the spores, and the spores are called zoospores. And they have a flagellum or flagellae, they have two, that enables them to swim. And so when you think about, oh, it swims, then it must need water in which to spread. So it does, it needs about 12 hours of saturated conditions, and that can just be water in between soil crevices or sitting on the soil surface. So the zoospores will swim, and this is showing a seed, but they'll swim to whatever plant part or root is available. And typically we see it right at the soil line along the crown of the tree. So they're giving off exudates, and the zoospores swim along. They can detect their host, and they don't need any kind of wound to cause an infection at all. They could just land on the surface and penetrate right into that plant with a specialized hypha. And then once it penetrates into the plant tissue, the phloem, the cambium, it kills the tissue as it feeds. So essentially it's killing the roots, but it can also be girdling the base of the plant. So this is what you might see. Sumac on the left, juniper on the right, where the plants will die, kind of like I said with pine wilt, very quickly within a few weeks of infection. So a lot of times when I hear someone contact me and say, oh, their tree or shrub died seemingly overnight, then this is one thing that I might suspect. The other thing about Phytophthora is that, like I said, it needs that saturated soil condition. So that's overwatering, maybe irrigation leak, but it can also spread by root to root contact. And so if you have two plants growing in close proximity, one of them is infected, there's a chance that it can spread to the next one, even in dry soils, without the presence of water, but through that root to root contact. And that's what's happening here. This cherry tree, there is root to root contact between this neighboring tree. It's turning yellow, starting to die. So some other symptoms could be a little bit slower death, where there's a thin crown appearance to the tree. The leaves are very small. They may turn red early in the late summer, and they also may emerge late in spring. And so in those cases, you get this kind of earlier dieback rather than that kind of overnight dieback that I already mentioned. In either case, if it is suspected, the tree is not going to be able to survive. Eventually it will die. And so one way to tell if it was Phytophthora that killed the plant is to go to the base of the tree, scrape off the bark, and look for where the Phytophthora has killed the phloem. And so it's this kind of cinnamon brown colored tissue shown here against this healthy cream colored tissue. So that's the margin of growth of the fungus. And of course, it goes on beyond down below soil line as well. So I mentioned the confounding factors are saturated soils, water, over irrigation, where water is sitting at the base of the tree for hours at a time, or in some cases where you have plants that thrive in arid conditions growing right next to areas or plants that need a lot more moisture, like turf. And so turf gets watered every day in some cases, but junipers don't need water every day. But here's where you may have a problem where they're getting the same amount of water. Along the same lines with the Phytophthora root rot is another disease caused by Phytophthora called bleeding canker, where it just shows up as these, looks like stains on the bark, causing these individual cankers. And this is out of Nevada. So Nevada, several years ago, Department of Ag there was discovering that their silver maples and some other maples were dying back. And they found that they had a lot of this oozing on the trunk, and scraping the bark away, saw, okay, the tissue is dead underneath, and identified it as a Phytophthora bleeding canker. So with the confounding factors for bleeding canker, it's water hitting the trunk constantly. Again, the same idea where the turf may get watered every day. And if the sprinkler is hitting the trunk of the tree for that time period every day, it's going to make it more susceptible or more possible that these infections could occur. What's happening is that the Phytophthora is in the soil, but the splash of the water is splashing the spores up onto the bark, causing those distinct little infections. So water hitting the bark and compacted soil where rainfall lands and it just bounces right back up onto the trunk. So those could lead to possibly bleeding cankers. All right, so management of these two diseases is to look for those symptoms, which is the wilting or off-color foliage, again, that late fall color. And a lot of times there's what we need to do to manage them is cultural, and it's prevention. So avoiding saturating those roots in the trunk. Any compacted soil, reduce that, add organic amendments. And then there are a lot of species that are completely resistant to Phytophthora. So if it has been detected, don't replant with the same species because it can survive in the soil for 3 years or more. So replant with one that's going to be more resistant. And then with those cankers, there has been some work on killing them individually by excising out, meaning cutting out the bark around that canker edge. And usually these cankers are pretty small, about 2-3 inches by 2 inches. And then on the canker itself, using a spray. This is foliar spray, but a spray directed right at the cankers here. Agrifos mixed with pentrabark applying them to the canker itself to slow the spread, prevent formation of additional zoospores. This agrophos could also be used where Phytophthora has been identified in the area and you want to protect the neighboring trees. So it's not something that's going to help a tree that's already been affected, but the neighboring trees from that root to root contact. All right, so another one that we're seeing off and on is bacterial wetwood. And this is one I know you're all familiar with as what we also call slime flux, where on cottonwoods or elms, you have a big old pruning cut and you have this ooze of bacteria that mixes with bacteria in the air and forms this kind of toxic mess, I should say, that drips onto the ground, kills the turf down below or other plants growing down below. And it doesn't always negatively affect the health of the tree. It's more aesthetic and things growing around the tree. So we're familiar with that. And we also call that bacterial wetwood. So just to explain this kind of slime flux, it's a mix of bacteria that naturally occur in the soil that enter trees that are susceptible to tiny little wounds, colonize the tree. And what happens is as they are metabolizing, these fermentation products build up within the tree, pressure builds up and it has to be released somewhere. So it gets released at branch crotches or pruning cuts. The bacteria comes out and then, as I said, it mixes with bacteria in the air. And these are some hosts that are susceptible. But what we've seen in the last 10 years or so is kind of more significant symptoms with bacterial wetwood that go beyond just that slime flux, where the bacteria are actually colonizing the xylem and going into the phloem tissue as well, and seriously preventing the flow of nutrients and water and causing actual tree death. And you don't really see that symptom of the slime flux coming out of a big pruning cut. The symptoms are a little bit different. They are these tiny, like the bacterial, the Phytophthora bleeding canker. So these tiny little areas of ooze on the outside of the bark. So here are some on this tree. And as I mentioned, it's killing trees in certain areas where this is occurring. So the scraping off the outside of the bark here and looking at the inside, the discolored tissue actually goes way into the tree, to the center of the tree. So it's not like the Phytophthora bleeding canker where the infections are happening on the outside and just killing the phloem. It's the infections are happening on the inside of the tree and the bacteria is moving its way out and actually getting to the phloem area and girdling and killing the trees. So this was kind of new for us in terms of the behavior of the normal bacteria that cause the slime flux were causing these symptoms in cottonwoods and poplars. Part of the problem for the greater incidence of this was where trees were growing in very compacted soil. They might have been near a water source that helped spread these bacteria, and also drought conditions can exacerbate the problem. In some cases, we saw a lot of flat-headed borers, suspected, well, are they moving the bacteria from tree to tree? And no, we believe it was just their feeding in the tree was, the bacteria were colonizing the feeding afterward, the feeding galleries after the fact. So for managing general bacterial wetwood, good pruning practices, make sure you get those cuts that are going to heal up nicely. the bacteria need a little wound in. And so if you have a mowing operation, you're constantly mowing over the roots, that's going to create wounds and allow for introduction of these bacteria. Trees that are actually under optimal watering and nutrition are a little bit less susceptible. So definitely we have found that nutrient deficiencies can increase disease incidence. And have a good diversity of plantings where you don't have that root to root spread from one plant to the next. For the regular slime flux, drain tubes are not recommended, but you can kind of clean out the area of the dead bark and the dead limbs. Okay, so thousand cankers disease. This is one that's actually been in Utah for 2 decades or so. But this last summer, we had a lot of reports of new infections or new cases where English walnut trees were not doing well, whereas we hadn't had so many reports in a long time. So 1,000 cankers of walnut. This is another one that's the pathogen is vectored by an insect. So the pathogen is called Geosmithia, and it's vectored by walnut twig beetle. And this is another case of the vector and the pathogen are both native, but there's been some things that have happened that have caused this kind of explosion of spread, not just in the West, but all the way to the East as well. So Arizona walnut was a native host, and so it's theorized that the beetles moved from the Arizona walnuts to other walnut species, and that they also hybridized as well. So this is a map on the lower left of the little red areas show where walnut twig beetle occurred. And you can see this is about 100 or more years ago, okay? And there's a little population in California, 1960s. And then the map on the top is where the walnut twig beetle is now. And that's December 2014. So it's a wider distribution now. But the point of this map is to show that that little population in California in the 60s, what they're theorizing is that it hybridized, with the population in Arizona and Mexico. Twig beetle that was able to go to more walnut host species and also able to carry the Geosmithia pathogen. So the disease, 1,000 cankers, showing where it occurs in the West. And it has spread, as I mentioned, to the East, where a lot of native black walnuts are grown. And so they're very worried about it in the East. So the twig beetle is teeny tiny. Here you can see the size comparison with the tip of a pencil. And as the beetle feeds, it creates a gallery in the tree, but it's carrying the pathogen with it. And so the fungus then grows out of that gallery, causing a small canker around the edge of the gallery. But when you think about, if you know about bark beetles in our forests, they attack trees by the hundreds or the thousands. And so it's the same with the walnut twig beetle, and hence the name 1,000 cankers. So each little twig beetle feeding in the tree causes a canker, then a ton of them are there. And so you get this massive cankers killing the trees. So here's just an image of some beetle galleries showing where the fungus has grown out of those galleries, staining the wood. And as the beetles attack the tree, the cankers grow and they kind of all coalesce into one. And this whole process on the black walnuts was found to take anywhere from 3 to 5 years. So it's not a quick death for the trees. Okay, so here, this was done by a plant pathologist in Colorado. So they took 2 logs, from an infected tree, put them in an enclosed container, let all the beetles emerge, and then collected the beetles in the one vial. So if you guys are wanting to guess, you can type it in chat. How many beetles do you think are in that one vial? The vial is about this big. So if anyone wants to put a guess, how many? 100, 1,000, so 3,000, 250. So you're all way too low. You'll be surprised to see that it's over 23,000 beetles. So that's just from those 2 logs. Yes. And so we have these beetles in Utah. And as I said, we were hearing some more cases of infection this summer. So the initial symptoms to look for might be some wilting in a certain part of the tree. Those wilted areas will then turn yellow. So we call that flagging. And then eventually the tree will start to die back. And the vial that was collected, that was done by a plant pathologist out of Colorado State University. And so I'm sure he had a grad student counting those. So susceptibility. Black walnuts are the most susceptible. And this is a lot of hosts here on the lower axis and the number of cankers shown in each bar. So black walnut shown here, and then butternut, which we don't really grow in Utah, is next susceptible. And then here's our English walnuts, are kind of in the middle. But we are seeing these become affected. So I'm wondering if this is from 2013, needs to be updated or looked at a little bit more. Pecan and hickory are not susceptible. This is a picture of a property in Richmond. These 2 black walnuts died over a period of several years. And this tree on the right is a Persian or English walnut that is now dead. Now the exit holes, so when a tree is suspected to have 1,000 cankers or something's going on with it, you would want to look for these exit holes. And they are tiny. There's a picture on the right shown with a penny. And on the English walnuts, a lot of times the bark is a little more smooth, so it's a little easier to pick out those exit holes. On the black walnuts with the real ridged bark, it's a lot more difficult to find them. And typically in the inner part of the ridges is where you'll see those exit holes. So if you look at this picture, study that inner ridge area, and there's about 7 exit holes right there. I'll just point, this one's obvious to see, but there's some others along here. Now, obviously this is a very close up of that bark. So we just want to keep our eyes out on the other walnuts in Utah. This is not a picture taken from Utah. This is out of California, but it's just showing the stained area of the bark on English walnuts. And unfortunately is affecting some of those trees in their the commercial walnut groves. And this is not a reportable disease in Utah, so it's not regulated. But we do like to know in extension where it is and where it's occurring. All right, so management of 1,000 cankers, and it's kind of like all these diseases. There's not a lot of options for cures. So trunk sprays, injections are really not very effective against the walnut twig beetle. So the idea is to, if it's been found in the area already, some people could put up a trap. So there's a lure that would indicate whether the beetle is present or not. But then prevention of spread. And so trees that have been positively identified, and again, that's why we want to know too, so we can get a good identification. The tree should be removed to get rid of those beetles. Walnut lumber is valuable, but shouldn't be moved from trees that have been affected. And if the wood is chipped, then that will take care of the beetles that might have been inside the trees. Any more you want to know about Thousand Cankers? There's a great website, thousandcankers.com. All right, so I want to finish up with 3 foliar diseases that, so I've said here, associated with cool, wet springs. So we don't really see these that often, but in the last 5 years, there have been pretty bad outbreaks of these diseases 2 times. So again, something good to watch for as our weather and climate conditions just really change constantly. All right, so one of them is bacterial blight, and the disease name is called Pseudomonas syringae. And it is actually present everywhere. It survives as what's called an epiphyte. So it's just getting it what nutrients it needs from the air on a lot of different plant surfaces. And this picture on the right is a magnification of the bacteria themselves right next to a plant stomate. So bacterial blight affects hundreds of different woody hosts. And most commonly, I've seen it on lilac, ornamental pear, serviceberry, ornamental cherry. So it causes a shoot and a flower blight. And these are some examples of the symptoms of this shoot blight. So on the far left is sweet cherry, lilac in the middle, Manchurian apricot on the far right. On the lower left is snowball viburnum, a red twig dogwood in the middle, and serviceberry on the lower right. So you can see the host range is pretty wide, but the symptom looks very similar throughout. Again, it's just these succulent plant tissues causing what looks like frost damage. And how it does remind you of fire blight, but I rarely see this disease on apple. So that's one thing to keep in mind. But I wanted to mention how the disease, or how the bacteria cause disease, is that they're able to turn water into ice at temperatures above 32 degrees. It's called ice. They have a protein called ice nucleation. And so they land on the plant surface, find a wound. They need a wound to cause infection. And then they, again, cause water to turn to ice. That kills the plant tissue. Then they feed on the ultimately dead tissue. So bacterial blight management is a combination of cultural and other methods. So plants that are nutrient deficient are going to be more susceptible. And plants that may have had powdery mildew the prior year are also going to be more susceptible the following spring. And lilacs are very susceptible to powdery mildew. And thus, I see a lot of bacterial blight on lilac. Pruning some plant species that are marginally hardy in fall or winter can also increase their susceptibility to infection the following spring. So those are all kind of factors of precondition that can cause plants to be more susceptible. But another way to prevent infections is don't prune on a rainy day. So prune anything out, all infections on a sunny day. And you don't have to prune too far. Where the bacterial infection occurs, the symptoms that you see, you don't need to go too far beyond those symptoms. But if this is a perennial problem on certain plants, then application of copper just before bud break could reduce the incidence of infection. All right, so another disease that we see kind of spike up and down is aspen leaf spot. I know some of you have areas where you see it every year. So it's a fungus, Marsanina is the name of the fungus, and aspen, cottonwood, and poplars are susceptible. So it causes these brown spots that, initially start very small with a little yellow halo around them. And they'll continue to grow and then merge into blotchy areas, and eventually some of those leaves may drop from the tree. So that happening year after year certainly does decrease the vigor of those tree species. So managing aspen leaf spots, those particular trees that are growing very close together, the humidity is raised. So thinning dense clumps is helpful. Avoid any irrigation from landing on foliage. And then trees, again, that are affected year after year can be helped by a fungicide application. So the timing is at bud break, but then probably it would need to be repeated 2, maybe even 3 times, spaced about 2 weeks apart. And chlorothalonil, one of the inexpensive options, is very effective. But some other options include products that contain azoxystrobin. All right, so finally, anthracnose. And again, we're thinking about this times of cool, wet springs. So there's several different species fungal species that cause anthracnose. And in Utah, we see it on sycamore, oak, and maple, but it can also affect ash and elm. And each one of those has its host-specific fungal pathogen that affects those species. But what we see are these irregular blotchy lesions on the leaves. However, on sycamore, the pathogen affects the wood as well and causes cankers in those infected areas. So on those cankers, spores will germinate in the spring and cause infection to the new foliage as it emerges. So with sycamore anthracnose, it's a lot more deadly because it's systemic within the tree. And as infections repeat year after year, you get what is known as witch's broom symptom. And so in the wintertime, you can easily see which sycamore trees are affected by anthracnose constantly, because you can see these witch's brooms pretty easily. So in periods of in periods of time where the environmental conditions are just right, then we have seen sycamore trees completely defoliated. But they will send out a new flush of foliage. It's kind of hard to see in this picture. This brand new flush of foliage actually is becoming infected again. And this is where there's real prolonged cool, wet conditions, even after the flush of new growth. So I've never seen this disease on redbud. If there is a case where you have a redbud tree that's being slowly dying back, it might be something else that's causing the problem. I mentioned anthracnose on maple. So here are some symptoms where initially little chlorotic, puckered areas that then die and turn brown. And that's maple, and then oak on the far right. So gamble oak is a very common host that we see it on in Utah. So managing anthracnose, you really want to watch for those new lesions as they start up. It's good to know if it's present or not, and maybe you can get a fungicide application on as soon as you start to see those lesions. Look for the small cankers on sycamore. And those can, if you're pruning a tree, you're up high in the tree, can prune out those localized infections on sycamore. But there are chemical options for management. Gamble oak and maple, again, those are not systemic infections. So they're only going to be happening from leaves that have dropped from the tree the prior year, the debris, the fungus sporulating on those leaves and bouncing up to the foliage. So on those trees, you could consider a foliar application, and the timing's at bud break. So as soon as that green starts popping out of the buds, and then repeating it 2 weeks later. So there's some options shown there for you. And for sycamore anthracnose, I mentioned, a systemic problem. And so in that case, it's going to get anthracnose every year when the conditions are right. And I forgot to say that when temperatures reach about 80 degrees and it's dry, spread of anthracnose stops completely, okay? But sycamores, you consider an injection. So the injection, of course, you're causing a little wound, but it lasts up to 2 years and it's very effective. Expensive though. So you'd want to save that for the specimen trees that you're managing. And so there was a question about raking up leaves. Certainly, yes, that's going to reduce that inoculum potential, but there's no way you're going to be able to get all of the little leaf parts that may harbor some of the overwintering fungal parts. And so raking is going to help a lot, but won't be the one answer. All right, so if you're ever finding anything you're not sure about, don't forget we have our diagnostic lab on the USU campus in Logan, the Utah Plant Pest Diagnostic Lab, and prices range for diseases from $10 on up. And so they may plate out plant material and see what grows out. They may do some kind of molecular or other types of analyses or assays to see what's going on. So don't forget the Utah Plant Pest Diagnostic Lab or even your own county extension office.